Hi guys, my name is Tim O'Keefe. I am the Science Department Chair here at St. Louis U High, and I teach anatomy, physiology, genetics, and also a biology course. What I wanted to share with you today is just a simple lab that we do in class and something that you can probably do at home as well. So what we're going to do today is we're going to extract DNA from strawberries. So what I have is I have just a few strawberries. You can either get frozen ones or just get strawberries from the produce area, and you're going to take those strawberries and just put them in a nice Ziploc bag. I want to kind of go through the materials with you. You want to also have some like household shampoo or something like that will work. Dish soap probably will work as well. You want to have some salt or meat tenderizer. Either of those will work. And you're going to make a solution with about half shampoo and half warm water, and then just add a fair amount of salt in there, and it will work. And then just stir that up, and that is going to be our DNA extraction solution. And I'll kind of talk about what that'll do in a little bit. You also want to have some 70% isopropyl alcohol. Remember, this is poisonous, so obviously you do not want to drink this. If you get it on your skin, that sort of thing, it won't hurt you at all. It's used as an antiseptic, but it's really important that you do not drink this. We're going to use that later in our experiment. You want to have some ice. You can just have ice in a bowl or something like that. I have some ice packs that I'm going to use for this. You want to have some sort of Ziploc bag. You can either use like a little snack bag or a little sandwich bag, which what I'm using here. And then you want to also have a cup, maybe some sort of Dixie cup like this. Have a couple of those. You can also possibly use something like this as well. So either of those will work. And you want to go ahead and make sure you have a coffee filter as well, because you're going to use that for filtration. The first thing you want to do is you want to put your strawberries in the bag. And you're gonna just physically break those up. So what I do is make sure kind of the excess air is out of it so I don't pop the bag. And I'll just take those strawberries and you just kind of squish them up as much as you can physically. What we're doing there is we're breaking all the cell walls of the strawberry and kind of getting a lot of the cellular material kind of out in the open. And once you've squished that up for a little while, what you want to do is, I usually like to stir up my shampoo, water, and salt solution. And then you want to just pour a little bit in there, maybe like 10 mils, but you can just pour a little bit in the bottom so it kind of covers the bottom of the bag is enough. You don't really have to measure it. You don't have measuring stuff at home. So you have something like that, right? And then you want to go back and you want to break that up even more. And I'll share a copy of this lab online as well. And the idea when you're doing this is you're mixing the soap or the shampoo with the solution, and what that's doing is it's breaking down the lipids, and the cell membranes of the cells are made up of lipids and proteins, and so that soap is going to break up the lipids. The salt or the meat tenderizer is actually going to help break up the proteins, right? Because the idea here is we want to be left primarily with DNA in solution. So after I've squished that up for a while, what you want to do is you're going to put that on ice. So in your bowl of ice, or I have a couple ice packs here, I'm going to put that in there and I'm going to leave that sit for, you want to leave that sit for 4 or 5 minutes. So once you've had the strawberry solution on ice for 4 or 5 minutes, you can pull it out, maybe just break it up a little bit. But what we're going to do next is we're going to filter it. So you take your handy dandy coffee filter that you get, and then just push that down into your plastic cup. And it's good to kind of fold it over a little bit, just keep, and pop it down in there, and you can kind of overlap it a little bit in there. And then what I'm going to do is I'm going to take my solution, I'm just going to pour it carefully through the filter so I don't make a big mess in mom and dad's kitchen. And I'm going to kind of hopefully get most of that out of there into the filter. It doesn't have to be perfect, but I want to get a fair amount of it out of there to work with. And then what we do is I'm going to grab the filter by the top and kind of just pinch it together. I'm going to hold that up, and what will happen is the fluid will start to drip through. And what I usually do is gently kind of knead it or squeeze it. Same deal here. One of the biggest things that happens to some guys is if they squeeze this too hard, they'll pop it and it'll break open and all the seeds and cell walls and all that will go through. You want to just get fluid in your cup if possible. And so you want to drain a little bit out of there. Kind of notice I'm gently kind of just rubbing it and squeezing it without squeezing very hard, because I just want to get fluid down in there. I don't want to break it. Okay, I feel like I've got a fair amount in there, so I'm going to put this to the side. What I'm going to do is, at home, you can just use a Dixie cup, is fine if you have one. I have some test tubes here, so what I can do is I'm first going to go ahead and just transfer the solution into the test tube. I'm going to pour it carefully in there. For you, you can just pour it carefully into your other, maybe smaller cup. You could probably even do this in the cup you started with if you need to, right? And there's just my strawberry solution that's got, mostly just the fluids from there. So it's got maybe some protein, some lipids, some DNA that have all been broken down. And then I'm going to take my my isopropyl alcohol that has been on ice. It's really super cold, or it's been in the freezer, right? And what I'm going to do, and remember, this is poisonous, so you're not allowed to drink this. What I'm going to do is I'm going to carefully we don't want it to mix together. We want the alcohol to sit on the top. So what I'm going to do is I'm going to carefully pour this down the side of the tube. And I'm going to pour about 10 mils, right? And then what will happen is the DNA is going to start to collect at what we like to call the interface between the strawberry solution and the alcohol. And if you have like a little wooden stick or skewer at home, if it's a little coffee stick, or sometimes like we use these skewers to make kebabs at home, if you have one of those, once again, you got to leave it sit for a couple minutes. But what should start to happen is the DNA should come out of the strawberry solution and it should collect kind of at the bottom of that alcohol solution, what we call the interface. And so once I've let it sit for a while, I can see that there's a white it almost looks like snot, like a white kind of snotty material in there. That is actually the DNA from the solution. So what I'll do, if I can, is I'll get my skewer in there and I will start to pull it out into solution. This will work. You'll be able to see this better on the close-up, but I'll start to get that DNA solution and it starts to stick to my skewer, and I can maybe pull it out of solution a little bit. I can see some DNA on there. And that is how you extract DNA from strawberries. Hopefully that was helpful to you. I think it'll be a fun lab for you to do at home. I really hope to see you guys at St. Louis U High someday. I think you'll love it here if you decide to come here.